Very good afternoon to all, and thank you for the opportunity to present our work here. I am ARAVIND Roy. I am a cornea faculty at L.V. Prasad Eye Institute. So our present study deals with the human ocular surface microbiome. When we talk about microbiome, what do we mean? So any body surfaces, the skin, the gut, or also the oral or the nasal mucosa is teeming with life. Some are commensals, some are pathogenic, and there is a dynamic interplay between these organisms. And when there is disease, the upper hand is by the pathogenic organisms, and when there is health, there is the upper hand by the commensals, which are the protective bacteria. Now we know that it has a significance in life. Is there a significance after death? Yes, probably there is, because we are seeing increasingly fungal infections in post-transplant, and this is also true because there are more lamellar surgeries, more DMEX, DSEX that is happening over the last decade. And another interesting thing is that the tissue that is coming to the cornea surgeon is preserved in a solution which is like MK is the most common, and also the others are like our the cortisol or usol, etc. And they are all having antibiotics. They don't have antifungals. Whereas after COVID, the Eye Bank Association of India changed the rules of cornea collection from not only 6 to 8 hours, but up to 24 hours of death, provided their body has been preserved. So with increasing time, a hot, humid cavity like the conjunctival cul-de-sac is an ideal breeding ground for fungus. And are we even understanding what colonizes the conjunctival cul-de-sac? And this was the background of our present study. This is a more of a basic science study, and here we collected the swabs from the conjunctiva of the cadaveric eyes prior to collection, prior to installation of povidone iodine, and we analyzed it for the types of organisms that populate it. So when we analyze microbiomes, there are two methods by which it can be done. Number one is the amplicon sequencing, where we target only very specific taxonomic markers, 16S, 18S, ITS, and then there we understand that these signatures are very unique for say fungi or one particular type of fungi, and these sequences are matched to the database. The other is the metagenomic sequencing, where we read the entire genome, but that is slow and more expensive. So most studies, including our studies, performed the amplicon sequencing. Then this was the methodology of next generation sequencing. Take a swab. From the ophthalmologist point of view, take the swabs, extract the DNA in the labs, perform a PCR amplification of the amplicon, which is the ITS2, and then match it with the library, which is already existing, and identify what are the pathogenic, what are the commensals that are there. Then there are some quality control indices which tell us that this is, for example, the alpha diversity plot, which basically means that this is a very diverse collection of organisms that are there in the samples. And in our study, we included all those suitable donors between 18 to 80 years who were refrigerated within 6 hours of death, and the collection was performed within 24 hours of death. We excluded all the known contraindications for eye donation, such as septicemia, drowning, then any other contraindications, exposure of the eye, road traffic accident where there is a contamination. So those were out. So we only took the healthy eyes. From there we took the swabs prior to collecting the cornea after instilling povidone iodine. For this study, 6 suitable donor eyes were studied, and we performed the OTU. OTU is the ocular taxonomic units, meaning that what are the taxonomies that are there in this microbiome, and then we assigned this to phylum and genus level. And the commonest phyla were Ascomycota and Basidiomycota. The commonest genera were Aspergillus, Bloomeria, Cutaneo, Trichosporum. So these were the common genera. The other fungi that we noted were Candida and Malassezia. So if we see, these are the heat maps. Basically, these bars tell us how much of each genus is present. So if we see, this is the blue colors is Aspergillus, and then we have the Coleotrichum, and we have some Candida also. So predominantly there was more Aspergillus after death. Species heat map distributions are again very similar to the genus, obviously. And just to give an overview of how this is just the microbiome of only one sample. And here we can see the tremendous diversity of the microbiome that is there in the eye after death. So here we see, if we just, and this is just the fungal microbiome. So here we have Ascomycota and Basidiomycota in red and green, and the yellow is interesting. That means that in this sample, 29% of the fungi are unknown, unclassified. Even we do not know currently with our databases that this much of organisms are not even known to us. So that shows how important it is to understand the microbiome and understand that these can be pathogenic, these can lead to possible disease. That was the purpose of the current paper. And from the phylum that we found, that Ascomycota was predominant, Aspergillus is commonest, but there is also good presence of Candida and Malassezia. So thank you very much for your patience here.